Hello and welcome to the Superposition Guys podcast. My name is Yuval, and my guest today is Clarice Aiello, faculty at the UCLA Electrical Engineering Department. Clarice and I spoke about her work in quantum biology, nature-made quantum sensors and their potential macroscopic biological impact, explored the possible influence of weak magnetic fields on cellular function, a future where personalized magnetic fields could be used for specific health needs, and much more. We hope you enjoyed this episode. Hello, Clarice, and thank you for joining me today.
Thank you for having me, Yuval. I've recently become a fan of your podcast, so I'm happy to be here and to be talking to the quantum enthusiast.
Wonderful. So who are you and what do you do?
I am faculty in electrical engineering at UCLA, trained as a quantum engineer. I work if you're curious, in the past, I worked with technological quantum sensors. I am sure a lot of people hearing this podcast knows, but you can actually prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. So that's what I used to do up to, say, 6 years ago. But at some point, I realized that nature itself was producing sensors that all the time overperformed humankind-made sensors. And it turns out that there's evidence that some of those sensors are bona fide nature-made quantum sensors. So I work in a field, an emerging field that is called quantum biology, that aims to establish the extent to which endogenous or native quantum mechanical degrees of freedom in biology influences macroscopic biological function. I'm an experimentalist, instrumentalist by training, so I build machines to study and control those degrees of freedom, quantum degrees of freedom in biological matter, in the same way that you would control those quantum degrees of freedom, say, inside non-living matter.
How about an example? So, what biological process do you think, or does the academia think at the moment is influenced by a quantum process?
Okay, I am sure you and a lot of folks here might have heard about the poster children of this field called quantum biology. Two very famous poster children of this field is photosynthesis and birds. So I don't care about photosynthesis, I don't care about birds, but they have the big advantage that they shifted this conversation from biology and chemistry to physics. So photosynthesis works better than any humankind-made solar cell. And now there is correlative evidence. I'll talk a little bit about the evidence in a second, but there's correlative evidence that it works by employing some type of noise-assisted quantum process in which phonons actually help the transport of the energy of the photon that absorbed by plants from the place where the photon is absorbed until the place where this energy gets dumped. The second poster child of quantum biology is, again, birds. Birds have been known for more than 50 years to navigate using, at least as a partial cue, the magnetic field of the Earth that is known, which is like orders of magnitude smaller than the magnetic field produced by your cell phone. So today, the standing hypothesis on how they do this is by employing a type of, and I'm going to be repeating this a lot of times, by employing a type of electron spin-dependent chemical reaction. So if that's okay, I'll explain a little bit what I mean. In test tube chemistry, there is absolutely no doubt that that's what's going on in certain chemical reactions. It has been also undoubtedly known. So here's the deal. In certain chemical reactions, there's a chemical reaction happening, and at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction effectively looks for the electron spin of a particular electron, and if the spin is up, or when I talk to physicists, I say if the spin is measured by the environment to be up, the chemical reaction continues through one path. If the spin is down, or measured by the environment to be down, the chemical reaction continues through another path. importantly, the macroscopic final products of those two distinct paths are also distinct. So there is no doubt that spin degrees of freedom, very finicky, actually influence the macroscopic final products of a chemical reaction. Now, if at the point that this chemical reaction is at this crossroads, this all-important electron spin sees, interacts, senses a nearby magnetic field, there happens a process that is effectively indistinguishable from what quantum physicists would call quantum sensing. The electron spin relies on superposition to sense this magnetic field, and this magnetic field will alter the probability of the spin being up or down. It's in this sense that the magnetic field, via this quantum knob, also can alter the probability that one chemical path is taken or the other one. And whereas there is no doubt that this is what happens in test tube chemistry, the cool thing is that birds, for example, seem to respond to magnetic fields in a way that is consistent with this type of spin-dependent, electron superposition-dependent chemical reaction being active under physiological conditions inside the birds somewhere. Right? And one cool thing that you need to know before I stop talking about birds is that it's well understood in the spin physics of those chemical reactions in test tube that the magnitude of the effect of the magnetic field being sensed or the magnetic field being changed paths is not monotonically increasing or decreasing with magnetic field strengths for reasons that are well understood and that for the experts have to do with like how the external magnetic field compares to nearby hyperfine interactions of this electron spin, just for the expert. The magnetic fields that can actually alter the macroscopic fate of chemical reactions are relatively small, on the order of the magnetic field of the Earth, on the order of the magnetic field produced by your cell phone. This also means that if you put the magnetosensitive proteins inside, like the 3 Tesla of a magnetic resonance imager in a hospital, the big 3 Tesla is not going to make a difference. The magnetic fields that make a difference are relatively small. So, and the idea is that then birds and other organisms in general are reacting to magnetic fields to the extent that they are reacting to the different physiological concentrations of products coming from these electron spin dependent chemical reactions. Now, I don't care about birds. All I care is that for more than 40 years, there is a wealth of, unfortunately, not very systematic data that shows that cellular function is macroscopically altered by weak magnetic fields in a way that is also consistent with this type of spin-dependent chemical reaction going on. And this ranges from DNA repair yields, regulation of cellular oxidants, regulation of cellular metabolism, regulation of cellular respiration, up and down regulation of cellular proliferation. So what I care about is trying to understand deterministically this quantum knob that we might have in this spin degree of freedom that happens in a wealth of different electron spin dependent that seem to be happening inside cells, and understanding whether we can control those towards some sort of altering physiological course, for example, for future therapeutics and the like. So this is basically what I do. Oof, that was long.
I want to make the distinction between the microscopic and the macroscopic, because when you started talking about chemical reactions at the atomic level, that's where quantum effects happen. But then you spoke about birds that, of course, much larger objects.
Yes.
How is that connection made between an electron spin in a bird to the bird knowing where to navigate?
That is the problem. It's very hard to study that. So evidence nowadays exists in test tube chemistry, and the next level of evidence is like for birds, for plates of thousands of cells, for like little organisms. There is no unambiguous confirmation or refute. There is just correlative data that those things seem to be driven by this type of spin-dependent chemical reaction under the hood. For example, in the bird, there's an idea of what the protein that can sustain this type of spin-dependent chemical reactions might be, but it's very hard to map this causal network, right? How does a spin-dependent chemical reaction that is probably altering protein conformation at the micro level, how does that translate to birds going right or left in the end? It's not known, and that's a problem, right? So this is why in our lab, what we're developing are experiments that bridge those two length scales. We are still building the experiments, but where we want to go is trying to start studying this type of spin-dependent chemical reaction, say inside a single cell, and at the same time read out, by a technique that I can explain in a second, read out how spins inside cells react to magnetic fields at very brief time scales, and how this spin preparation translates downstream into like cellular differences in cellular physiological phenomena using regular fluorescence microscopy techniques that people in biology have been using for decades. It's like in the same experiment being able to initialize, manipulate, and read out spin degrees of freedom. And once we learn how to do this, map out how those things are connected. How if we prepare a spin in a particular way, manipulate with magnetic fields, we can manipulate or not in a way consistent with this theory, things that happen at the cellular level. So our experiments that we were building, they really look like they're glorified microscopes with coils, but they are synchronized down to the nanosecond. So it's really what you would find in a quantum sensing-like lab. But our sample is biological, right? We work with proteins, in the near future with cells, for example.
The magnetic field of the Earth is fairly constant, certainly in direction and magnitude. So I have two questions. One, do you recommend in your home, do you have a weak magnetic field because it's helpful? And the second one, how does the dynamic behavior of the field, in your opinion, change the impact?
So then again, it's crazy. That's the point. The answer to your question is, it's crazy. So first of all, the magnetic field of the Earth is not that constant. The magnetic field of the Earth, I think it varies between 30 and 60 microtesla, more or less, and that already makes a difference. And birds also seem to track the direction of the magnetic field. So in the model, and again, those things you can model with tools from quantum physics, like the tools from open quantum systems, in very simple ways. And those models already account for what happens in chemical reactions in test tubes. And there is a dependence on the model on magnetic field direction, frequency, and intensity, right? The other thing I want to mention is that if you grow cells, grow stuff under very good Faraday cages, you mess up with biology big time. So I have a friend, his name is Peter Vierlinger. He is a researcher at the TU Munich. He's like a bona fide precision measurement physicist. What he does is, for a living, he builds very good Faraday cages with all sorts of like passive, active compensations. And what then he does is he shoves ultra cold atom experiments inside, because you don't want like the magnetic field of the Earth to alter those experiments. So he started hearing about those things of like, magnetic field effects in biology. So biologists convinced him to do an experiment in his lab. So here's what he did. So in his chamber, so magnetic field of the Earth about 50 microtesla. Inside his chambers, DC level of magnetic field about 1 to 2 nanotesla, so very, very small. He grew tadpoles for 2 days under and this has been reproduced by other groups, and this seems to stand. It's really, really crazy if you see the data. If you grow tadpoles, like frog embryos, for 2 days inside those chambers, One with a control where you apply a tiny magnetic field of the Earth, like the Earth inside. The tadpoles after 2 days are okay. In the other experiment, he grows the tadpoles just under like nanotesla level magnetic fields. You see like 30 to 40% of embryo malformation, in that the embryos get all deformed and are no longer viable. There's other research saying that if you grow cells without the magnetic field of the Earth, you change epigenetics, you change like this type of like biological process involved in how cells evolve or something. I'm not sure I'm the best person to explain epigenetics, but one seems to be changing physiology big time, and this opens up all sorts of questions, starting with maybe space exploration, right? So can you reproduce in Mars? Can you do space farming in Mars? The magnetic field of Mars is very weak. So how do we do this? And that's something that I've actually been trying to tell NASA for some time now, that it's not only microgravity, it's not only like cosmic radiation. We need to be thinking about different magnetic fields. And magnetic fields. And no, I would not be worried about magnetic fields from the environment. So here's the deal. The magnetic field specificity seems to be very large. Like each chemical reaction depends very precisely on being tuned by a certain magnetic field frequency and intensity, right? So it would be very bad luck if your cell phone manufacturer really hit that sweet spot to either make your cells behave worse or better, right? Which is the crazy part. By just changing a small magnetic field, you can up and down regulate processes. Like you can either increase or decrease the speed of cell proliferation. And this is already being deployed by some companies that have found non-deterministically some type of like weak magnetic field that seem to magically, for example, reduce the spread of one type of cancers. Like the most famous of these companies is Novocure, that produces Optune. This company is now valued at $10 billion, and they have found a fortuitous magnetic field that happens to downregulate to actually decrease cell proliferation rates in a very specific type of cancer, in a way consistent with this type of spin-dependent chemical reaction. It's really crazy because like magnetic field intensities involved are tiny, and it seems to work, and it's valued at $10 billion.
How prevalent is this field of study of quantum-related biology? Are there thousands of researchers around the world, or just a handful?
So it's growing. I hope your viewers won't see this as a heresy, but I think that quantum biologists today where quantum computing was, say, 30 years ago. There was a lot of 30 years ago in quantum computing, there was a lot of theory and not a lot of groundbreaking experiments. I think that right now it's sort of the same. There's a lot of quantum theorists involved in research in quantum biology, but up to now, almost all the experiments have been performed by biologists and chemists. Those experiments are hard, they're beautiful. So imagine like taking every migration season, taming like 20 birds during migration and messing up with the magnetic field. Those are hard, right? Those are beautiful. We should respect those type of experiments. They're necessary, and I'm claiming they're not sufficient. I think that the field will advance once we start applying bona fide quantum-like experimental techniques to try to control those quantum degrees of freedom in biological matter in the same way that you would control a spin in diamond, right? So, and there's a lot of people in this field. There have been for, about for 30 years or so.
If you're successful in the lab and the research that you're doing, what do you expect to be able to achieve, say, in 5 years?
In 5 years, I would like to be able to either establish or refute down to a quantifiable noise level the extent to which spin phenomena are like weak magnetic field related to spin phenomena can alter cellular function, either saying, yes, we can see that it's the spin doing this, or saying, no, down to this noise level that is measurable, we don't think it's causal. Because I think that this will move the needle, this will make people pay attention, because all the data in quantum biology, for all the flavors of quantum biology, and there are many, it's all correlational. It's either data from chemistry, that there's no doubt there's quantumness there at room temperature, or there's data, macroscopic data that behaves in a correlative way. So where I want to be in not in 5 years, but maybe in 25 years. So I want to have an app. I'm serious about this. I want to have an app that you can go there and, like, today you need help with wound healing. For example, wound healing depends on magnetic field in a way consistent with this type of spin-dependent chemical reaction. You go to your app and say, I want to do wound healing, and then you click and your phone produces the exact magnetic field intensity and frequency that you need, and then you go with your phone close to your skin. So the technology to produce those magnetic fields they're already there. You don't need anything else than a cell phone, because all you need is the correct strength and frequency, and strengths are weak on the order of those produced by your cell phone. What you need now is the deterministic, if you will, quantum code book on which magnetic field influences which chemical reactions. And for the expert, I'm maintaining that what you need is the knowledge of the local spin Hamiltonian to influence each electron spin degrees of freedom for each particular protein, depending on what the protein is and where they are in the cell. And for this, you do need those big optical tables, those big synchronized, beautiful quantum experiments. But the end product is easy. The hard part is finding this code book.
As we get to the end of our conversations today, I wanted to ask you a hypothetical. If you could have dinner with one of the quantum, or I guess biology greats, dead or alive, who would that person be?
Wow, that is okay. I used to like Feynman a lot. Now Feynman has decreased a little bit in my priority scale. I would like to have dinner maybe with Schrödinger to inform him of how the world has evolved. So right now, when I teach my students, I no longer call Schrödinger's equation because Schrödinger was a bad man by a lot of criteria. I teach my students the evolution equation. So I would love to talk to Schrödinger to raise awareness of some of the bad things that he did, and that to show him that the world is evolving into other more healthy directions towards people who study science and especially minorities in physics.
So Schrödinger, not Tesla.
No, no, but good one too. And I think I would like to. Okay, good one too. I would like to have dinner with both Feynman and Schrödinger and make the same exact comments about minorities in physics.
Very good. Clarice, thank you so much for joining me today.
Thank you for having me.